RNAs are not important. But now we know that non-coding RNAs are also important. And another definition is called the DNA sequence of replicates, ignoring the epigenetic variation. Like I was showing, if you see last 2 decades, the sequencing technology has drastically evolved. Including the cost, rate, everything. In 2001, when we were talking about the ABI, today we talk about the lots of technology, Illumina, Solid, Bio, IIMT, and which has reduced drastically the cost to the output. And this results in the generation of huge knowledge, which presents that new opportunities which we can use. And as I said, that cost has gone drastically down, and it further going down, and we are able to, now we talk about the genome of $1,000 or these different million. Even first genome came for more than $1 million. So like I was mentioning, a lot of genomes are available. This technology, like I mentioned, technology has become very cheap. So sequencing is not a big deal, but sequencing a genome is not enough. Why? When you have one cultivar, you will get the data repetitive of entire one genome. It is unique to its thing. Another genome becomes, so now what you get, there's a lot of overlap, but still there are some unique things. And when becomes the third thing, that you will keep on increasing, you will keep on getting this thing. So this is the concept called about the pan-genome. And that is sensible genome, which is the overlap between 2 or 3 cultivars. And here comes the core genome, which is conserved across all the expressions which are being studied. So I'll explain this thing in coming class, and this presents the basic also of pan-genome. What is a pan-genome? Like I mentioned in the previous slide, it represents full complete diversity within a clade original all genes. It represents entire diversity of that genome. It represents, like I was showing the previous slide, it doesn't represent the genes of one variety or one cultivar. It shows the full set of the genes present in that species or clade or the population. First pan-genome came in 2005. It started the microbial, and the concept of pan-genome was developed by Detlin group in the paper published in 2005 in PNS or the research paper. What they did, they took 6 strains, representing 5 major disease-causing genes of Streptococcus aggregatia, and they developed 8 genomes with 8x coverage. They took the whole genome alignment, and the gene similarity was determined using those things. And the first pan-genome was the first concept of pan-genome was developed. What are the basic features of pan-genome? As I mentioned, the core genome, sensible genome. Core genome are made up of genes that are present in all axes. Like I was showing the graph previously. If you have 10 cultivars or 10 extensions you are studying, the genomic region which is conserved in across all the 10 genomes, and it is called the core genome. Then another concept is the dispensive genome. Some genes which are common in 2, 3, or 4, but not in the all, it is known as dispensive genome. And there is another concept of the orphan genes, which are the line-specific, sex-specific genes, which are found only in particular species. If you see from this diagram, accession 1 represents the orphan gene. This is the core genome, diverse core, like I said, core genome present in all the 3. Dispensive genome present in 2 or not others, and orphan genome is present only. So this shows how the pan-genome concept was developed in 2005, and it evolved with the time. Different components, different evolutions of theory came. I'll explain those things. So now coming to what types of pan-genome are available. One is the reference-based pan-genome. So what here do that all the reference which are accessing the sequence, they are mapped to a reference and to develop pan-genome. What is the pros? What is the benefit of this? It is very fast and easy, and very easy to enter. It's a small file, you can take it in your pen drive and you can do it. But what are the negative issues? It doesn't present the entire component of the gene. So what we are doing, we are eliminating reference. If reference doesn't have that gene, it will not represent. Like here, if you see expression 1 has those things, expression 2, now from that, if we align it, start aligning it against the reference genome, we are going to miss this orphan genome. The reference doesn't have it. So our pan genome will look something like this, and it will miss that orphan genome. So while this is fast and easy, it has that it will not be represented entirely. A lot of pan genome were so far based on the reference genome. Okay, I think I have been asked to fast enough. Recently, we came up with the concept of super genome. So instead of doing the reference-based genome, those things, what we do that our position is that we develop a genome in the individual species. You have that definition from one species, and then have the species genome, and then altogether we develop super genome. Coming to the chickpea work analysis, like most of you are aware of it, Dr. Badal is there, so he will tell you more about chickpea. I'm not going to explain, there is more those things. The 13 group and it is at the leadership value came, he developed this draft genome, 13 members there, so he's part of the paper, so he'll explain more maybe. Last year we developed the input genome assembly using the, together with the IC and other mapping approaches. Like I said, sequencing single genome was not enough. So what we did, we started to do a lot of sequencing, and in 2019 we sequenced more than 400 chickpea sessions. So a total of 8.8 terabyte sequence data generation, which identified 4.9 million SNPs. It resulted in analysis of about domestication analysis and selection tree for grenade and set of origin of chickpea development. From this, then we started the bigger initiative of 3,000 chickpea genome sequencing. It was one of my biggest projects when I was in IIMT together with you. So what we did, we sequenced more than 3,000 chickpea samples. About 30 terabyte data was generated, 4 million identified. Last year, this paper came in the Nature, big story. So using this data, what we did that we identified about 4 million SNPs, including 33,000 cultivated and about 200 wireless registrations are there. Here we presented the chickpea genome. So what we did that we used the chickpea reference genome of series frontier, and another reference genome was this genome. We use the wild chickpea wild reticulatum, and then we use the data from the cultivated data from the cultivated excision and wild excision. And if you can see, we developed chickpea genome of which 532 came from the reference genome of series frontier, additional 2.93 came from the basic genome, and 53.9. Hello? Want me to finish or I should close it?
ना वो कुछ पढ़ना चाह रहे पढ़ अभी नंबर काम कर रहे हैं
We could not see any of your slide action start to present. We are running short of time, so very good.
So just I'd like to close it. And now we are going to the general compensation report. In that, I'd like to thank all the collaboration, including Dr. Mahendra and Dr. Madhav was sitting there, and all the work we did at IIMT with the strong collaboration of the education partners. Thank you.
Thank you.
Hello.
Thank you, Manish, for a nice talk. Do we have a question from audience? One or two quick questions, especially from students.
Hello.
If not,
Hello.
we thank Manish for his nice talk and enlightening us about the QTL mapping. And
I'm not, I think there is issues of I may leave that for right?
thank you. Thank you.
Now the second topic presentation is from Dr. Kanwardeep Singh from USA, alien gene transfer for targeted crop improvement. Is it available? Okay. Boys, it will be quite difficult for you to understand about genomics. In fact, genetics itself is a very tough topic, and we don't know whether farmers are ready to accept our genomics or they have their own genomes to research be conducted by themselves. मैं खुद मानता हूँ कि हम सबसे ज़्यादा वैज्ञानिक जो है वो किसान ही होता है. He knows what to do and what he wants, and we should follow the vision of the farmer. हमें किसान को पूछना चाहिए कि वो क्या चाहता है.
If there is an issue, then we can switch to speakers. You can have Dr. Bush first. Later on we can.
Dr. Bush,
Yes, sir.
where your presentation said present and future prospects of floriculture in India. Is online? Has it come? Okay.
माइक दे दो.
तो आ गया, ये इन्हीं का है. Welcome, Professor Bush, a man from floriculture. ये रंगीन आदमी है. फ्लोरिकल्चर का आदमी हमेशा रंगीन होते हैं.
First of all, I welcome to all of you, and I congratulate especially to the IIMT University to invite me as a speaker regarding the present status and future prospects of floriculture. So first of all, good afternoon to all of you, sir. You know, the floriculture is a very fascinating subject for all of us. So before going to the floriculture, why we adopt the floriculture? We know what is agriculture. Agriculture is the production and processing of crops and animals for human use and environment. It means in agriculture we do with our animal science. So how to make a good environment or how to make the sustain the environment, then you switch over to the horticultural crops. So in horticultural crops, we are dealing with so many crops like fruits, vegetables and other things. But floriculture is one of the very peculiar thing. By that way we can make our farmers maybe more prosperous. As Dr. Nazir said, we think over on the farmers because the farmers is the backbone of our country. So next slide please. Next slide. You see the farmers mostly we have a more or less the small and marginal farmers. You know, the very small land holding the per capita is 0.12 hectare. It's a very big one, and below 1 hectare and 1 to 2 hectares are small and marginal farmers. So we think over that, how to make them the prosperous. So what is the solution for that? How to make the farmers prosperous? So these are the some. What is the solution then? So adoption of new approaches. We have so many approaches, so many technology. By that way, we can do in the farming community and increase their productivity. The innovative methods of farming, then increasing diversification in crop choice, that is high value crop. We know the floriculture is a low volume, high value crop. So in the community area, we grow the crops and we get more money. We think over the from 1,000 square meter area, we can say this is the 1 unit as recommended by the National Horticulture Board, we make a poly house of 1,000 square meter area that regarded as 1 unit. So from that unit, we can get the approximately gross return from project to 10 lakh rupees, and ultimately we get the net return approximately 6 lakh rupees from 1,000 square meter area. But the question arises how to manage the money to the farmers. The question is still there. So enabling environment that support innovation, then market-driven production, domestic and export. So we think about the both domestic as well as export. So most of the loose flowers are mainly grown for the domestic purpose, and generally the high quality export cut flower is for the export purpose. So in that direction we can adopt the business. Either we go for the loose flower or cut flower, depending on the demand of the locality. Then increase in productivity of crops, we can go for the vertical farming. You know, in the city areas we have a concrete jungle. So in that city areas, we can go for the vertical farming. We can grow the floriculture unlike other horticulture crops like fruits and vegetables. We can grow on the terraces. So by this way, we can make a good environment and also giving the more prosperity to the even the Indian family. Then improvement in efficient of input use or cost saving technology. Then increase in crop intensity. We have developed so many the high yielding varieties, and they are good responsive to the fertilizer and irrigation. So by that way we can increase the productivity of the farmers. Then improve price relation by the farmer that can be achieved by quality maintenance according to the global standards. So we must know what are the different criteria for the selection of the varieties for different flower crops. As you know, in the gladiolas, we require at least 1 meter length of spike length. That is the minimum standard for the export of the gladulous cut flowers. So like that we can think over that, what are the different criteria. We thinking that criteria, we grow, depending on that criteria, we select the varieties. So next one, this is the floriculture. There are different advantages of this is the aesthetic horticulture. Then we also include in the culture and tradition. We know that we just require the flower from the birthday to the death. Every time we require flower. So it is in our culture and tradition. Then this is the money spinner. As we already told you that from 1,000 square meter of poly house, we get the 6 lakh rupees as a net return. So you can say if you spend 1 rupees and you get the I think hardly from cereal crops the 1.25 or 1.4 or maximum 1.5 rupees. So the kind of benefit cost ratio is 1:1, maximum 1.5. But you think about the floriculture, it giving the more than 2.5. Okay. So it is dependent on the different situation and different crop varieties. So and ultimately it is giving the more employment. You know, from 1 acre of land, you require hardly 1 or 2 labor for the growing of the cereals like wheat and rice. But you think about the floriculture, it requires at least 10 labors per acre. It means that giving the 4 times labor, that gives the good opportunity as a job, for the job point of view. Even the young person that can also work as in the packaging of the floricultural crops. From the packaging point of view, they can pack the flower in a packet at their own home, if they belong. So, but for that people, we can also give some employment. Then ultimately, if we have the more money, then we are happy. Next slide. Hello. So the present uses of floriculture in India that give the beauty and aesthetic value, these are all already discussed, environmental value, employability and aromatherapy and nutraceuticals. So medicinal aromatic plants are also included in the floricultural crops. So we can go for that. Even the, if you think about the marigold, so marigold also, the powder of marigold we can use for the more yellowing of the yolk color of the poultry. Okay. Even the different cosmetic industry, we are using the different oil like rose oil or jasmine oil. So then what are the demand? Demand for flowers in India. So for the traditional flower, there is steady demand in religious purposes, personal development and home decoration. At that time, they required steady demand, and these are the seasonal demand, the religious ceremonies, festivals and marriage. There are so many festivals like Diwali festivals, like Holi festivals. At that time, we required the special types of flowers. In the modern days, the modern flowers, mainly from the institutions, they require the main flowers demand. And the seasonal, at the time of different occasion, we require seasonal flowers. Like Valentine Day, we require the red rose. At that time, the price of the red rose is very high. Even then, one cut flower of the roses goes up to $11 per flower. Next slide.
There was a film Red Rose. Rajesh Khanna. Have you put up the look? Yes, sir. वो फिल्म जरूर देखिएगा आप। The Red Rose. And Red Rose को उस फिल्म में बताया गया कि उससे राजेश खन्ना पागल क्यों हो जाता है। Nehru was wearing red rose or buttoned red rose in the coat.
Yeah.
That impact was positive for him, but in Rajesh Khanna, in the film, red rose was negative impact on him because of his direction was different, which was not accepted by that flower. ये उस फिल्म में दिखाया गया, देखिएगा वो फिल्म में फ्लावर्स का क्या रोल होता है. This is to be studying about that.
हाँ, एक वैरायटी आपकी रोजेस में फर्स्ट रेड वैरायटी, that is a very leading variety. So the employment, next slide please. Then we just go for the interpretation of it from substrate to waste, all can be being exploited. Next. So these are the major sectors, different sectors. You know, we grow for the roses for the loose flower, cut flower, then we go for the business for the seedling plant production, then bulb plant production, then we go for the dry flower. The 60% exported earning from the dry flower. This is the one business. Then we go for the rental services. If you grow the crops or even the landscape plants in pots, in big pots, so that pot plants can you go give the as a rental, give the plant on rental basis. In the Kisan Mela, we are taking the giving the rent of 10 rupees per pot for 1 day. If one stall is demanding for 10 pots, it means that giving the 100 rupees per day. And there are 4 days Kisan Mela, 400 rupees they are getting from one stall. And there are so many stalls out there. So by this way, there are different occasions we are just providing the pot plant on rental basis. जैसे आप देखते हैं, आप हम रेंट पे लेते हैं, आप टेंट लेते हैं, कुर्सी मेज सब लेते हैं, तो इस हिसाब से हम इसको भी एज ए रेंटल बिजनेस यूज कर सकते हैं. Then we go for the flowers for the extraction of oil. That can be used by the different cosmetic industry. In that direction, we can grow the flower. Then even the tissue culture. By the tissue culture, we can grow, propagate thousands of plants. You know, in the Jarvera cultivation, nowadays the people want the tissue culture plant. And one tissue culture plant, the price of one tissue culture plant is 36 rupees. If you develop the tissue culture nursery plant, then you are getting more money. In the, you can see the one company is there, the agri genetics, like Seal Biotech. They are developing Zervera plant. So by that direction we can think over that. We can install the tissue culture plant and grow the different planting material. So one of the major bottleneck for the increased production of the floriculture. The people want the elite plant of material, and that is not very much reliable. Okay, the farmers want the elite plant material. So by this way, we can provide the elite plant material to the farmers. Next. So one of the industry is nursery industry. The different vegetable crops, we are just grow for the seed production of vegetables. In that manner, we can also go for the seed production of floriculture crops. The flower crops are very highly perishable in nature. If the marketing problem is there, suppose in the Uttarakhand area, there are very hilly areas. So from that hilly area, there is a marketing problem. So we can grow such type of flower, and those flowers which produces the seeds, like the winter season annual flowers. So we have 100 types of winter season annual flowers. So winter season annual flowers can be grown in our Indian condition, and such type of flower we are very, very difficult to grow in the European countries. At that time, there is a lot of snowfall. They are growing the flowers in the glass house. So that is very, very difficult, very cost involving. So by from the low cost, we can grow the flower in our Indian condition. So this is the one aspect of nursery industry. We can go for that. Next. Then another one is the cut flower. So in the North Indian condition, we are growing the roses, cut flower as well as rose flower. Even the carnation, or the leading crop. Then gladiolus. This is the major and high class bouquet we can made by the gladiolus spike. So nowadays it is very much demanding. In the Lucknow region, you have seen most of the farmers are growing the gladiolus, and they are exporting the gladiolus directly to the Gulf country. So the farmers in the Lucknow region are very much curious about the growing of gladiolus. One or two farmers are also just approach me, and I just giving the approximately 6,000 to 10,000 gladiolus cones to the farmers, and they are very happy. So by in that direction we can think over that. The institutions can also grow the different elite planting material and supply to the farmers. In that direction we think over that. So other crops like the Gentidaceae, Anthurium, orchids, such crop we can also think over that. The Gentidaceae can be grown as a monopoly crop in the temperate climate. It is a very highly potential temperate bulbous crop. Nowadays the people want the new thing. They are saturated with the roses, chrysanthemum, gladiolus. They want new flower. So such type of flower we can incorporate in the floriculture industry. So one of them is Gentidaceae, and even tulip, like lilium, we can introduce and we can adopt for the export purpose. Next. Then the loose flower, there are different uses of loose flower, garland, mani, gajra, loose flower. When in the North Indian tradition, if you just inculcate the loose flower in our culture, then the demand will be high. As you see in the South India, the flowers are in culture. So every woman want flower in South India. So such type, जो भी हो, they are very religious.
No, it's not religious. We have Gajra because of what?
Yeah.
I will tell you.
Okay. So next. So teaching, research, training and extension support unit, we can also develop such type of unit, because the farmers want the new technical person. They want new technology. So how to deliver the technology to the farmer from lab to land program. So by that way, we are bound to establish so many institutions, so that the human resource person we can develop, and that fellow can provide the some technical person to the farmers. I think the the different KVK, Krishi Vigyan Kendra, we have the only the horticulturist or agriculturist, but no any floriculturist. Okay, so from this forum we said that the at least one floriculture must be there at every Krishi Vigyan Kendra, so that we can give the best person to the farmers. The grassroots level we can provide the technical person. Okay, then next. Okay, next. So you see this is the photograph. We just designate the low volume, high value crop. You see the left hand side there is a fruits, and the right hand side there is a flower. So we think over that this is a low volume, high value crop, and we think over that. Next, the different data is available. The share of the area is only 1.24% in horticulture crops, but their export trade is the 4.25%. If you think about that, they are giving the more money as compared to the fruit crop. Next. Hello. So the facts and figure about next. So we have the very meager amount of the export 0.2% to 5%. Even then we have the share of the Indian condition India in the world market less than 1%. So we think over that. अभी बहुत रास्ता बाकी है, अभी बहुत करना है. Next. So these are the nowadays we are exporting the cut flower in terms of weight, not in the number. So this is the figure how many numbers of cut flower in 1 ton. If you think about the 1000 kg, so glandulous crop the accommodate the 8000 spike is equal to 1 ton. It means approximately 100 gram to 125 gram per spike. So according to that, you have to think over that how much charges are taken by the airlines. Okay, so think about that. Next. So these are the share of the Netherlands 33% and others are 10%. So we are in 10%, less than 1%. Next. So these are the demanding of the leading demand of the world market is roses, then carnation, then chrysanthemum. So according to that, we think over that which flower is more demanding in the export market. Next. So what would be the strategy? So diverse agroclimatic condition. So depending on the climatic condition, we grow the crops of different locations. Like in the hill areas, we go for the carnation. In the South India, we go for the roses, like Pune and Bangalore condition, there is a mild climate. In this condition, like Meerut, we go for the roses, then gladiolus, and then the Jarvera. Such type of crop you can grow here. So skilled manpower, nowadays some skilled manpower are also available. So by taking the advantage of skilled manpower, we can grow the floriculture crops. Next. So these are the what are the times for the demand. So from January, September, October, November, December, and January, February, this is a high demand for domestic as well as international. So at that time there is a highly snowfall there. So at that time there is a high demand. So we can export the flowers at that time and get more money. Next. So next. So these are some flowers, cut flower like gladiolus. You I think might have seen. Next. Then roses, these are the then carnation. The special type of techniques required carnation is a very brittle in nature. Okay. Then the lilium nowadays in the Uttarakhand some peoples are growing lilium, and approximately the cost of one lilium bulb is 30 rupees they are importing, and the total cost of cultivation from one bulb is one spike is 25 rupees, and they are selling the spike of 60 rupees per spike. So they are just double. One fellow is working at the Devi Dhura region. Dr. Manoj is there. They are supplying the lilium directly to the Pune. Next. So these are new crop, Alstroemeria, the right hand side. Then these are Alstroemeria, the name of the varieties. We are at Hamanai was at Bharsar. We are growing the such type of varieties, Alstroemeria. Next. Then the loose flower, you go for the rose, jasmine, chrysanthemum, chrysanthemum. Chrysanthemum is only for the South Indian condition. Next, these are ladies' pouch flowers. Such type of the annuals flowers can be grown to beautify our landscape. Ice plant is the ultra dwarf annual. You can landscape the plant. The cornflower, this is a big national flower of Germany. They can also incorporate for the landscape purpose. And sweet labelium. Next. Then this is the calendula, or known as pot marigold. We are also using as the medicinal purpose in the Ayurvedic, the homeopathic. Next. Then cut greens. These are the cut greens. Nowadays they are selling the cut greens in the Delhi market and fetching more price. Next. Then in the flowering house plant. Nowadays the city areas they are demanding the house plant. So one even the household ladies can adopt such type of business in the city areas. Next. Then foliage indoor plants. These are the hello. Yes. Next. Then floral crops and cut flower arrangement. This is another business. We can think over that. Next. Okay. Next. Bonsai. So, one of the very special types of training required. If you want to training program, you can contact with the National Botanical Research Institute, Lucknow. They are giving the training on bonsai. So by that way we can develop your own enterprises. Next. There are different types of bonsai. Next. These are some example of dry flowers. The flowers are very papery in nature. Okay. Next. There's some wild ornamentals. We have the Valley of Flowers in Chamoli district. So such type of wild planting material can be used for the domesticate or the commercial purpose. Next. The landscape is also one of the major aspects. We think over that. Nowadays, every people in the city area, they want the good lawn. For every garden, the 75% beauty comes from lawn. So you can say the lawn is the heart of the landscape. Every people want the good lawn. Okay. So nowadays, demanding of the lawn. If you want to make a lawn, so at least you require 1 kg lawn grass for 1 square meter. Okay, and they are charging the 25 rupees per kg of grass. So according to that, you think over that, you can develop your own lawn. So this is the nursery production of ornamentals. Next. Yes. So if you have the 2000 square meter area, it means a half acre of land. So how much you get the money? the net profit you can get the 5 lakh rupees from half acre of land through nursery. You can develop the nursery as a business. Next. Okay. So these are the standard of the glandulous criteria. Then economics of glandulus. Next. So this is the year-round production of the marigold. Marigold can grow either throughout the year. So just extreme hot and extreme cold. You just skip them, and every month you grow the roses. So by this way, you can supply the marigold flower to the market and get the money, and if you have a loan, to return the loan to the bank. So by this way we can make a good credit. So such type of thinking must be think over the how to make the continuous supply of flower to the market. Next. So this is the next. Yes. Backside. This is the net return of the roses. This is the one study of the Tamil Nadu one fellow, and they are giving a very good data. By this way, the return is 6 lakh rupees from 1,000 square meter area in the protected condition of roses. Next. Last one. This is annual flower production. Yes. Yeah. Back. So annual flower production, if you see over the calendula, you see like that. At our Nagar condition, we are costing the 5,000 rupees per kg of seed. Okay? And you think about if calendula is 400 to 425 kg seed per acre. So it means it is giving the 20 lakh rupees per acre. If demand is high and adopting the good practices, then we think about that. We can go for the seed production of flowers. This is the maybe the one of the major business. Next. So these are the temperature. Why we see here we require if you want to export oriented unit, you want to export the flower, you have a good refrigerated van facilities. So if there is a 1 minus 1 degree centigrade, by dry storage we can increase the waste life of the flower. So it requires a facility. So by this way, at the block level, we can make a facility for the refrigerated van. And Mawana is giving some subsidy to purchase the refrigerated van. So by this way we can promote the floriculture to the farmers. So these are the marketing channel next to. So these are the companies involved for the floriculture business. So these are the some constant. The first constant is the availability of elite planting material. So we think about that, we grow the elite planting material and give to the farmers so that the farmer may benefit more. And the finance is the major problem. You know that in the glandulus, we require for 1 acre of glandulus, we require 60,000 pounds. And 1 pounds costing 3 rupees. It means 1,80,000 rupees required for 1 acre of planting. So 1,80,000, is it possible for the farmers? No. So what will be our strategy? So the glandulus have the cormel, very small size of corms, that is known as cormel. If we grow the cormels up to 3 seasons, and after 3 seasons we develop their own corm. So by that way, we can convince the farmers to go for glandulus. So this is the way. Or we can make a self-help group, make a registration and take a loan from the bank. So by this way we can promote the glandulus. So help promote floriculture by joining hands with floriculturists. Thank you so much. Any question, please?
ऑडियंस प्लीज आप सेंड करिए।
हाँ स्टूडेंट आप पूछिए जरा, आपके मतलब की ज्यादा है इसमें।
ये पढ़ भी रहे हैं अभी।
आप कोर्स पढ़ भी रहे होंगे ना?
हाँ, पढ़ रहे
प्रोडक्शन
हैं।
टेक्नोलॉजी ऑफ ऑर्नामेंटल मेडिसिनल एंड लैंडस्केपिंग
पढ़ रहे हैं, पूछो
वो
भाई।
कोर्स पढ़ रहे होंगे जो आईसीआर ने दिया है। अच्छा डॉक्टर साहब से आप मेरा नंबर ले लीजिएगा, जब कभी जरूरत हो तो आप पूछ लीजिएगा। क्यों सर? हाँ, थैंक यू सर।
Thank you, sir. Shall we proceed to next speaker? Please wait. Yeah, please wait.
फूल आहिस्ता फेंको, फूल बड़े नाजुक होते हैं। खाना बनाया गया इसलिए बनाया गया।
Very nice.
Please message the speaker that he has to take care of time. Right? It's a routine 20 minutes and he should be 20 minutes.
बढ़िया। एक्चुअल में क्या है ना, ये मैंने पढ़ा ये सब नहीं सर, आपने हिसाब से जीन ट्रांसफर जेनेटिक्स आपने एक इसमें फ्लावर कप को लेके
Okay, so can I start? Okay, all right, great. I would like to start by thanking Ashok for putting together this wonderful conference. Holding the schedule of great competition. My topic, what I am sort of going to talk to you for next 15 minutes is my work on AUG transfer for targeted profit improvement, general background on why AUG transfer is important for crop improvement perspective. Is if you look at this upper box, which sort of represent your elite varieties or cultivars that farmers grow during the course of evolution, domestication, and breeding. If each of these blocks sort of represent a disease resistance gene, for an example, you could use other abiotic stress as an example. We have a very limited genetic genetic load among our elite cultivars that we are working with to develop future generation climate resilient cultivars. But in contrast, if we look at the wild variety of crop plants, they still represent a repository of genetic information that could be used to develop biotic and abiotic stress resistant and climate resilient plants. Keeping that in focus, that I've been working on for a while. Even though it is very promising, the biggest challenge that we face when we are looking to transfer genes from wide variety of crop plants is the presence of chromosome pairing and recombination barriers. If you see on the right side of the screen, this here I'm providing wheat as an example. So wheat has 3 genomes, A, B, and D. If you cross it with any of its wild relatives, whether it is rye or any other distant relative, in the presence of the strict chromosome pairing and recombination barriers, very limited recombination happens between the cultivated wheat and its wild relative. Whereas in the absence of these chromosome pairing and recombination barriers, we see lot more chiasmata or lot more regions where crossover happens and genetic exchange of material happens. To bypass these barriers, we do geneticists and cytogeneticists over a period of time have designed multiple strategies, which includes using irradiation or using nucleosidal genes, or even manipulating recombination using certain deletion mutants. They have been able to successfully transfer human genes from wild relatives. But the problem with those methods is that most of them have been not, they're very cumbersome. It takes a lot of time to initiate those transfers. And many of the transfers are still large transfers because of the smaller size of populations. So we have been working on to develop a new homologous recombination based approach which results in small reciprocal and balanced transfers. Again, just a brief overview or background of what is homologous chromosome pairing and what is not. So if you look at the interface stage, as shown here with these different colors, so these lighter and darker red chromosomes are homologous to one another, whereas they are homologous to the colored chromosomes. So in the presence of an accurate homologous chromosome pairing control mechanism, so the red will only pair with red, whereas the green will only pair with green to ensure that correct chromosomes come together and pair and exchange the genetic material to give you stable haploid gametes at the end of meiosis 1 and meiosis 2. We have been working to identify one such pairing control. So this is an example of silencing one such gene as shown here in the E. This work was the pictures that you are seeing are the work of virus-induced gene silencing, which is a transient nature of gene silencing, but this work has been validated using RNAi also, which is permanent form of gene silencing. So here basically what you do is you design an antisense or a caplex control to your gene of interest, you rub it on the plant and see what phenotype you get. So here all the above three are our positive controls where we see a normal chromosome pairing behavior, which is that we mostly see bivalent where two chromosomes come together to pair. But in the presence of gene silencing, we start seeing these abnormal, uh, where we start seeing these hierarchical chromosome pairing associations, which highlights that the chromosome pairing control mechanism has been positively disrupted. So then, using this gene, where we were able to induce higher order chromosome pairing associations, we wanted to see if we can further induce homologous recombination between the chromosome of wild relative of crop plants. So what we did was we used a wheat line that was carrying an alien arm, as shown here with these red colors. These chromosome arm come from the wild relative of wheat, and then we crossed it with a normal wheat line, and then on those F1s we performed virus-induced gene silencing using our candidate gene, and then throughout the F1s we did quantitative PCR, as shown here in this figure, for our candidate gene to find plants that show gene silencing. So if you closely look at this graph, so this is our negative control, which is F1 MCS, and then we sort of we selected three F1 plants, 8, 9, and 10, which showed about more than 90% of transcript reduction for our candidate gene. So then we took those F1s that were showing gene silencing, and then we backdropped it to another normal wheat line to develop a DC1F1 population. So this whole process, including identifying components, it took us closer to 1 year, which is significantly faster compared to any other method of making gene transfer and read, which easily takes between 5 to 10 years to initiate any kind of recombination between the right relative chromosome and the chromosomes. So then the next step that we did was we wanted to verify these primary combinations. The first approach that we used was a sequencing based approach where we used these primary combinations uh, and did RAD sequencing or restriction dilation based sequencing where we selectively enrich the gene proportion of the gene. So here, the simplified version of this slide is basically what we are looking to see is that if whether our primary recombinant plants carry any alien or carry any alleles that are coming from the wild DNA. So here, the red color sort of highlights the allele that is coming from the wild relative, whereas the green color shows the allele that is coming from the weak parent. So on the far right, you have these two controls where no recombination was observed. Whereas these were the plants that were generated using our approach or what we call transiently inducing virus-induced gene silencing, and we were able to recover plants carrying multiple introgressions from wild relatives. The smallest introgression that we were able to transfer was about 40 kb in size, and within that 40 kb there were only two genes that were coming from rice. So we are sort of highlighting the power of our approach that you could selectively transfer single genes from wild relative of croplands. Then we further wanted to study what is the nature of this homologous recombination is. So then compared to 5B chromosome in our population, we looked at this specific arm just to study how many single crossover or how many double crossovers we were able to see, as well as we wanted to study if the rate of recombination, homologous recombination along the arm of the wild chromosome was same or was it any different. So as shown here in this genetic distance graph, so you could clearly see on x-axis, this is the distance or the length of the short arm, starting with 0 MB and going all the way to 300 MB. That recombination goes up and down, which is generally assumed to be when recombination is happening in recombination hotspots and recombination cold spots. So homologous recombination also follow the trend of natural homologous recombination. Then we wanted to select ideal recombinant plant that because we were able to recover plants that were carrying small segments as well as large segments, we are generally interested in single gene. So we designed some markers. We got some markers from older publications also. So here we were looking for dominant as well as co-dominant markers to further screen this population in the next generation and see if those transfers were stable and they were passed on using simple Mendelian genetics as they should be. Again, here this is a skeletal map of one of our primary recombinant plants, where we were able to see that in the next generation, different segments were co-segregating as we expect for a stable transfer. Then the next step we used was we wanted to see if this gene is functionally preserved across monocots and dicots. So we did, although not shown here, we did the silencing of the gene in Arabidopsis also and saw high-order chromosome pairing behavior. Apart from that, then we wanted to study the functionality of the gene, what are the features that are common across monocots and dicots. So first thing we studied in detail was gene expression throughout meiosis. What we identified was that this gene specifically it highlights a meiosis specific expression as shown here in this first graph of neuron beat, where the gene expression is high throughout interface up till pollen development. And then when we look at ZMAs, we also see very high expression during prophase. And similarly, in soybean and chickpea, we saw highest transcript abundance in pooled meiotic tissue compared to young leaf, to young flower and mature leaf, highlighting that the expression regulation of this gene is preserved across monocots and dicots. And then we wanted to use this approach, or we want to characterize this gene to design these approaches or design this technology in dicots or other crops of the pollens. Before that, we are trying to optimize the transient gene silencing approach in dicots. We are using a DRB based virus system and diapers, which is a tobacco retrovirus. This has been successfully used in tobacco, Arabidopsis, and many other crop species. So we sort of try to use this approach in soybean to optimize it. It has been successfully shown in soybean also, but we are trying to optimize it in our lab. So first approach, so the first crop that we started with was soybean, and we used a PDS gene. Uh, the gene is a very good phenotypic marker that once you silence it, you start seeing photobleaching as seen here in these two plants relative to control, where the leaf sort of stays greener. So then after seeing the visual phenotype of photobleaching, we validated that data with transcript reduction and found that PDS silence plants that were showing phenotype of had significant transcript reduction. Now, shifting gear to understanding the context of crop improvement or using the saving gene transfer technology, we have to be very careful in terms of which wild relatives we sort of start with. So here we are giving an example of chickpeas, where we are working on Ascophyta blight, how we are identifying the wild relative that we are going to use in our crop improvement program. Uh, this is specifically for Ascophyta blight. In chickpeas, there is no immunity or good plant resistance in the cultivated germplasm for Ascophyta blight. So the only resistance that is workable is either present in Cycles reticulatum or in tertiary gene pool accessions. So here, as you see, that once we screened about 98 accessions of Cycles reticulatum, and we found only one or two that showed significant level or high level of resistance, which is workable. And then similarly, we characterize the tertiary gene pool of chickpea also for Ascophyta blight. And as seen here in the second graph on the right hand side, that we see certain accessions which are showing immune response. So these are the accessions that we will focus on to transfer disease resistance using our approach. So this is what this work is currently underway, but we are transferring the resistance or immunity from this wild accession of chickpea to the background of soya using our approach of truncating balancing using our immunologist control gene. Hopefully next time if we meet or get to talk, we'll be able to discuss about how to sort of transfer. And with that, we would like to thank all of our funding institutions and NSF, USDA, United Soybean Board, and USA Pulses. We are thankful to our project team for coming together to make this possible, and the Gene Shifter team also. And I'll be happy to take your questions, and I hope that I stay within the time limit. Thank you.
Thank you, Karandeep Singh, finishing in time. You hear me? Is there any question from audience? Yeah, yes, there is one question. Maybe you can come forward. So question is, what is gene silencing, right? Karandeep Singh, do you hear me? Right. So there's a student, she wants to ask what is gene silencing.
So whenever you do gene characterization, so there are 2 or 3 different approaches. So under normal circumstance, your gene make its mRNA transcript, and then that mRNA transcript codes a protein to give you a gene function. So when you are looking to characterize that gene, either you can overexpress something that you make more copies of that gene, or you reduce the number of transcript or mRNA copies of that gene in particular cell or a particular plant system. So that is what gene silencing is. We try to characterize a gene for its function by reducing its mRNA level in the system. So eventually the amount of protein is less. So the gene is not functional in that particular plant system or cell.
Thank you. Is there any other question? If If no, then we must thank Karandeep Singh for really nice talk. Thank you very much, Karandeep. Thank you. So with this, we proceed to last speaker of this session, Dr. C. Bhardwaj. Dr. C. Bhardwaj is a very senior scientist at IIMTU, and he is going to talk about a very interesting topic of translating, if I can read it, genes to varieties for drought tolerance in chickpea. Sir, and I can guarantee you he is going to finish in 20 minutes. That time will not include.
Honorable Chair, I think I really thank you for setting the debate of the day that the farmers know very well. They are the scientists and it is well upon agreed. But at the same time, the basic sciences pave the way. So once you understand the problem in the field, it becomes essential to look at. And way back when first Sir Ronald, before this started, Sir Ronald Fisher has started biometrical genetics. This was the basic question that the art of breeding was going to be removed from the breeder and it is going to move into the biometricians who are measurement scientists. It's exactly not so, friends. It is like this that you need to understand that these tools are to complement you. These tools are not to supplant you. Starting with this, I should be moving down into the thing where translating genes, I think I'll be more comfortable with that. I'll be more comfortable here down, translating genes to varieties for drought tolerance. Now, basically, this is a challenging question that appears everywhere, that with the changing climate, we have certain new challenges that the breeders, the biotechnologists, the biochemists, molecular physiologists, even the horticulturists, as a team need to face. That the abiotic stresses and biotic stresses are causing real harm and how we are going to provide resilience. Everything has a solution, but the basics needs to be worked into. And when we are basically looking, the whole of the pre-breeding which makes it longer is to make and enable the resources that are available with the breeder so that he can easily deploy these genes into the cultivated lines and then release as a varieties. So we have three phases, but we need to understand that initially it was exactly as I put it, it was an art of identifying the things. Then came the biometric genetics, where you used to evaluate, albeit phenotypically. Then you started moving in a way where you try to understand and co-localize these genes or mine these genes through biotechnological approaches or bioinformatic pathways. Once these have been identified, that's the translational approaches where you are going to deploy to lines that are easily crossable into your cultivars. And that makes the pre-breeding aspect, and it's a whole part of hidden science where Professor Mahendra Tudi, Varshmi and his crew, Manish Kivar, and including our colleagues at NIPGR are working. Unless we have these tools in hand and the precise way in which we can transfer them into cultivated lines, the problems of genetic drag do exist. These are tools, there are many tools, including genome editing to gene silencing to that of marker assisted breeding to the genomic assisted breeding to the genome introgressions. But you need to understand to have a clear-cut objective, where to start off, and have all the dots of the team and good funders in place. Now, if you look down, when we start looking at chickpea is the largest pulse crop certainly grown in India. Pulse means chickpea because we have more than 50% production, and it's the second largest in world after the kidney beans or the red beans or the rajma that you call it. But certainly abiotic stresses play a havoc here. Be it drought, be it heat, or be it salinity. And drought is one of the major problems that we are going to address, and there is this peculiarity, which is good in the most favorable condition, certainly does not fare well in unfavorable conditions because the genes that have been complemented to make it resilient to abiotic stresses or biotic stresses certainly pull down yield. And pulses over centuries have been growing in the marginal lands, not for now, for centuries along. And there has been a beautiful paper by Professor Bahel, which states that they have lost all their yield contributing genes for greater resilience to the various stresses. Now, if you start looking, so we started looking at the chickpea production in the Indian subcontinent, and we saw that the yields were greatly plateaued. The first thing was, can we break the yield plateau? And we started analyzing a global germplasm collection. This is only a subset that I'm presenting here. And then we could clearly see that the Mediterranean germplasm, whether it was wild species, whether it was the land races, that we basically obtained from the Mawana region, West Asia and the North Africa region, clearly had a branch away from the Asian region, and we predicted greater genetic gains, and then we moved ahead to check down whether there are any genes operating in the system. When you start looking at the abiotic as well as the normal, particularly in the drought stress conditions, now there is a peculiarity when you start looking at the cereals versus the legumes in general. If you see in cereals, there is a stage where the vegetative growth ceases and the reproductive growth commences. So higher harvest index was able to get gains in case of cereals, and this was the fundamental thing that they worked on in case of green revolution. But in case of pulses, which we have actually inferred, it is higher harvest index coupled with greater initial basic vegetative mass that plays a major role, because the reproductive structures basically are axial in nature. And then 8%, whooping 8% comes from the pod ball and then from the nearmost leaves. So we need to understand that it is the basic vegetative phase that plays a major role. We are lost on 2 slides, but it's okay. It's okay, that's okay. Now, if you are looking down, then when we started looking at these genes, we started looking at the way are any specific genes that are playing a role in case of drought tolerance. We have analyzed about 40 or 50 genes that have been identified by many workers like Spruti et al. From Vashne's lab, where Mahendra has come. And then we could actually hit upon 3 genes: dehydrin, abscisic acid, ripening transcription factor, sucrose phosphate synthase factor. See, when you look again at drought, it is conditional to that stage and to that locality. When you start looking at drought, particularly in case of chickpea in the southern latitudes, in the southern states, the drought starts somewhere after the summer solstice commences, somewhere, let's say, January 14, what we call in normal language there as Sankranti, and slowly the temperature starts rising. So the root basically has a phenotypic ability to respond to this changing drought. Now just compared to the North Indian conditions where we are, and somewhere in March 1st, suddenly the pole temperature transforms from 10 degrees to 15 degrees difference of higher atmospheric temperature. That means we need to also look into the above ground factors, maybe like relative water content, membrane stability index, how they work on. And as I said initially, one of my students, Manu, has worked on this to identify the gene groups. The second student, Dr. Tapan, who is now with ICARTA, started identifying the allelic variation in the DHM gene. This was one of the genes that was quite conserved across the pulse domain, giving resistance. And then we reported for the first time the ASR which we assembled into the assembly, please. And then we could identify that it was akin to the late embryogenesis abundant Lia proteins, which actually imparts drought tolerance. It's a constitutive expression, and most importantly, it's a novel gene which we have identified for the first time. Now, one more group, Dr. P.K. Jain, with whom we collaborate in the national project on transgenics and agriculture. we started looking up, as I said, with respect to relative water content, do you have any genes? And we could get this ethylene responsive factor gene, particularly that of 116. And we have seen that, which published by one of my students, Parmeshwaran, that there is a crosstalk of gene against various abiotic stresses, be it drought, be it cold, be it salinity. So it expresses, so this was one more that we said that if we are able to target one of the abiotic stresses through a specific gene transfer, we'll be able to do it. Now came the question, can we start off somewhere? Can we start basically of somewhere? And already Rajiv's group has identified a QTL for greater root volume, and can we combine this with the above ground traits that we are trying to do? So we developed a mapping population 4958 into 1882, where this QTL hotspot, we call this as a QTL hotspot because it almost has 12 traits. And then we started looking at fine mapping of this region because of paucity of time and just rushing down through. And then we could actually co-localize this to almost 13 candidate genes, which Mahendra would be explaining in detail. But then what was the most important thing is that we could identify 2 candidate genes, which were again localized to one specific candidate gene. We could prioritize the SNP mutations in these 3 genes from QTL hotspot in the paper that we have published in Nature, and then started the work that the Honorable Chair was saying, identification of genes is not alone the end answer to the farmer's problem. And certainly when you have a good team of across the discipline, each person doing the targeted and focused work, this is what we are going to achieve, that we could translate these genes into cultivated lines. We just did not release cultivated lines as genetic stock, but we released them as varieties, not through any AICRP or just state, but we have released at CVRC, not 1 line, but about 7 lines, not in India, but India, Kenya, and Ethiopia. That indicates the success of gene-based translational technologies. Dr. So what we basically tried to do was, since this QTL was identified in ICC 4958, we tried to progress into, mind you, when we were looking, we told Varshne and Tudi, hey, we just don't want to translate it into any variety. I'm basically a hardcore breeder. We said that we want to develop a variety that was already a mega variety. That means it has about 400 quintals of minimum breeder seed production. That means it's already a high yielding variety, well established in farmers' fields, and can we make that yield resilient against drought? And that's what we could do and achieve this. So we transferred it down at the bottom ground. You can see the Pusa 372, which is already proved to be high yielding, but its root structure, I'm going nearer to them, with its root structure, and that of the ICC 4958. And this was the QTL region, the QTL hotspot that we used using the foreground markers, transferring to Pusa 10216, Pusa chickpea 10216. Of course, we developed the fine mapping populations, we evaluated at 4 locations, we used the generation advancement technology there where we used it, IRA, Dharwad, in the final stages, the ICRISAT location also. So we were able to take 3 generations per year. Yeah. And we evaluated all these drought tolerant introgression lines for various physiological, biochemical, that is metabolomic parameters as well as yield parameters, screen them under the what we call as glass house conditions. Next please. And we could conclusive prove, not in 1 line, but DBG 505, DIBG 205 and BGM 10216, that yes, these genes that have been identified and the QTLs that are co-localized when transferred into mega varieties using the genomic assisted breeding will provide yield gains. And then this is a data over two years at the national trials at 22 locations. I've just taken the most drought tolerant controlled locations where you can look at the moisture percentage. At flowering, it is about 20, at maturity, it is about 15, a whole lot of almost 45 days. And the BGM 10216 out yields by almost whooping 11% and is the first MABC variety released in India. With this, we ushered India into the world map, proving that marker-assisted breeding technologies can give yield resilience. We just didn't stop there. Next one, please. These are photo edits. Just in last Jan, we already resiliented or insulated another mega variety, Pusa JG16. And the thing was, certain times we failed to appreciate our own Indian researchers. But Bill Gates all the way came from US to visit only 3 technologies in India. One, the energy conservation technology, which is done at Dehradun. 2, the COVID vaccine, which was developed at SACS. And 3, the drought tolerant MABC lines that were developed at IRA. We recently also are in line for this USA award. But whatever it is, that speaks the resilience of these technologies. But this technology is not produced by breeder alone. It has been produced by the whole gambit of this team who are credited in developing this variety. It's a disservice. It's what we call déjà vu. They do acknowledge and collaborators in the campus. We do acknowledge and collaborate them in my varieties. Mahinder and Varshne are also co-developers of our varieties. So we didn't stop there. We have gone with the Northwest Plain Zone release of IPC L4-14, Pusa 405, both of which were dedicated last year by Honorable Prime Minister Modi for the nation. Next. So that's what I'm saying. It's, as I'm saying, it's not just in India. The superiority of marker-assisted, genomic-assisted breeding, even at Kenya or even at Ethiopia, where Jiledu, the first high-yielding line in the background of JG11 was released. This speaks of the way that genomic-assisted breeding can take. So in brief, what I would like to state is that genomic technologies are not a bane, but a boon to the breeders. You have numerous technologies at our hand, the genome editing, the gene silencing, which the earlier breeder was saying, right? Then you have now coming over to genomic introgressions. Then we are moving down further ahead. How do we use them in crop improvement? One of the other aspects that my lab at IRA is doing is genomic selection technology, which Manish Rukiwal was saying, where we are trying to increase the total genetic grading and therefore increase the breeding value. But if we have certain genes which aid in this process, there is nothing wrong in using these technologies than staying aloof. Now you go to any of the farmers that Bill Gates visited in Latur, I can provide the names as well as numbers as well as the latitudes, because he has done this in 5 farmers fields in Latur and 6 in Bundelkhand, and then he wrote to the PM that these technologies next are a boon. Well, I say this is not my single work. We have Bharadwaj, Neeraj and Patel for this paper who have developed, Jane and Nimi from NAPB, Aditya, Suren and Yogesh. I am also the adjunct with University of Western Australia. For this paper, Rajiv and Manish, but certainly for the varieties, Mahinder, Preeti and Rafat. And of course, this work is what I represent of my about 18 students who have come through in this program. I thank Ritu and Dr. Ashok and all for giving me this opportunity. Thank you.
Thank you, sir, for fantastic talk. And it's really pleasing to see when years of your research work get translated into something which farmers can use. So thank you again, sir, and congratulations for this wonderful work. Is there any question for speaker? There are many. Great.
Ashil, please.
Thank you so much for your.
You are young. You can speak loud.
Yes, sir. Sir, thank you so much for your very fruitful lecture. So my question is that whether you have evaluated your tutorials or not, sir?
Yeah, this is the most important work. Basically, not contradicting anyone. We have already shown generally most of the papers earlier, quite earlier, were identifying the genes and publish, but no paper, no accepts without validation. But living out, I've shown you already that we have validated these QTLs at greenhouse level, at physiology level, at biochemical level, and also at field level. And of course, I've shown you one slide on that.
So what was the population size for validation?
This is very good. What is the population size for validation? Basically, when we are looked at the nils, we had a population of 150, but when we are looking at large scale validation, we are done in a 500 lines genome-wide population.
So as you are working on chickpea, suppose sir, I am working on wheat. So what would be the suitable population size for wheat?
This is not that. It is not on the size of the crop alone, but it is also the genomic reason that is basically important. Yes.
My question is also related to validation part. Like you have validated your all these genes or like through biochemical and various approaches, but you haven't talked about like CAFS marker. Is there any use or whether these are quite useful?
I don't put it that way. First of all, you need to understand what is the objective of your breeding program. The markers are very much useful. So if you don't understand what is the objective of your breeding program, it is like this, that if a ship doesn't know which port it wants to go, no wind is useful. So you need to understand that they're very useful markers, but in this we already have gene-based. The marker NAPGR21 was sitting right inside the gene. It was a SSR marker, so I could easily use it. Why should I go for a CASP where this can be done in just my lab? And it is very, very cheap. They're useful, but here I haven't used.
Yes, sir. I had few questions, but I'll discuss over the lunch. Right? Before she invites us for lunch, please do come back after lunch because we are going to have a very interesting lecture by Sir, and he's going to talk about how flowers affect the personality of a person. So he will tell you what kind of flowers you should buy and what kind of flowers. Thank you very much. Thank you all the speakers for this session.
Thank you, Bharadwaj sir, for very brilliant session by you. I think you have done a lot of efforts in field of gene variation, and you are doing a lot in this regarding this field. Now, I would like to invite faculty members. I would like to request faculty members or delegates and girls candidate for delicious lunch. Please move for lunch session. And there will be second session for